Hey mushroom nerds, it's Anna McHugh. I am having a lovely time at the North American Mycological Association annual foray, and I have been looking at mushrooms, attending lectures about mushrooms, talking with some of my mushroom friends spread from far and wide, so it's been just a delightful and delicious experience. I am delirious in a variety of other D words as well. One of them is dumbfounded by the number and diversity of species that we have found, even though the conditions have been reasonably dry. We're in the Appalachian Mountains outside of Asheville, North Carolina, near Hendersonville. And so I want to share with you just a couple of the things, in particular, this beautiful fish milky cap mushroom, also known as the corrugated milky cap mushroom. The scientific name here is Lactifluus corrugis. And I have a variety of specimens that kind of give you a sense of the whole range of how this beautiful mushroom sort of manifests itself. And this is a mushroom also I wanted to highlight because I don't find it at home, in Raleigh. So I am at a higher elevation. These mushrooms were foraged from a forest that was primarily rhododendron, but we also had white pine, we had oak as well present. And so, my understanding is that these mushrooms do grow in association with oak, but despite the fact that I live in habitats that are just teeming with oak trees, this is not a species that I typically get to see in my hometown, or at least the town that I call home, Raleigh, North Carolina. So I want to talk to you about this beautiful specimen, and then also just a little bit about diversity and sort of observational science. I would be really foolish if I tried to recap all of the things that I've learned, and it is not even done, but suffice it to say, attending a mushroom event with an association like NAMA, the North American Mycological Association, is an opportunity to meet people who are experts in different, very wide regions of the world, but also experts in specific areas of identification. If you're into wild mushrooms, which is kind of my interest, but we also have people who do, the culinary arts. We have basket weaving. We have, people doing DNA sequencing and microscope work. So it's a fabulous time to get together and sort of get a sense of the different, dimensions of the discipline of mycology as a citizen scientist, as a hobbyist, and also as someone who is a fancy pants, researcher who goes to fancy pants locations and finds fancy pants macrofungi. And when I say the word macrofungi, what I mean are fungi that produce fruiting bodies like mushrooms. So that's a good, sort of overall term to really include a tremendous number of different types of organisms that make, mushrooms of one kind or another to reproduce. Okay, so let's talk about Lactifluus corrugis a little bit because this is a good edible mushroom. Some of these are a little bit larger and more mature gilled than I would be interested in eating. Oftentimes when it comes to deciding when a mushroom is over the hill, there are a couple of things to look out for. First of all, of course, any sign of, rot or sort of slimy streakiness. That's oftentimes, some bacterial stuff happening, and mushrooms do spoil very quickly. But in the case of a lot of mushrooms, I am okay, as they mature, the gills tend to get larger relative to the overall density and thickness of the cap flesh. And at that point, you have spores going everywhere. It's certainly not like this is a problem. It's just that gills are a little bit more fragile. And so, in this particular case, I would probably leave this in the forest. And as I actually am handling this specimen a little bit more, I'm going to open it up and see if my suspicion is correct that it is full of bugs. Oh, okay. Hold on, it is falling apart. So the upper portion of it is nice and clean on the inside, as you can see. And so, if I did decide I wanted to eat this, I'd chop it up and I'd cook it thoroughly. But as we descend into this stem, it's kind of a little bit squishy. Oh yeah, here we go. This is the shot of all of the maggot damage that has come to pass to this poor little mushroom over the course of its lifespan, which is, not more than a day or two. Oh. So we also have fire ants in the mountains, I just discovered. So reporting to you live, there are all kinds of, occupational risks to mushroom foraging, even if you are at highly structured foray events like I am. So returning to this fruiting body, what you have with Lactifluus corrugis is a mushroom that's very aptly called the corrugated milky cap. And as you can see, it has this sort of brownish, but also like it's brown, orange, red, I would say, as far as its coloration. And but it has this really nice sort of corrugated surface to it, so it's a little bit wrinkly. There is another mushroom that I do find at home, which is Lactifluus volemus, also called, commonly the fish milky cap. It looks very, very similar, except it's like a more lighter terracotta orange, and it is more smooth, and it has like some concentric growth zones. But here you have a really distinct and pronounced sort of corrugated, appearance, which I just love, because even handling this mushroom is really fun to do. Another thing that you'll notice here, and it's really quite pronounced, is it's kind of pruniose is the term for it, which is sort of this like whitish patina that you see over the top of this sort of roughened surface. And, to that point, the mushroom is not like slimy or anything, and it varies in color from this sort of, this is a very light colored specimen, so it's sort of an orangey tan with a little bit of a sort of mahogany color. And then as you get into, darker examples, this is one that I put a little bit of water on so that you could really see the, darker coloration. It's almost like a dark, Pinot Noir red wine type of color on the top of the cap. And again, this very pronounced sort of rumpling. And even the young one, like if you look at it, you can see some areas are quite smooth, but there are spots of this warped sort of rumpling going on. So what makes this a milky cap is it produces milk when you damage it. And so Lactifluus is the name of the genus. And there are, I guess, 3 genera of mushrooms that produce milk of some kind or another when you damage them. And when I say produce milk, that's what I'm talking about. So both Lactifluus volemus, that other one I was talking about, lighter, sort of lighter orange, and Lactifluus corrugis, so more dark and corrugated and a little bit reddish, produce a lot of this latex. So it's just a juicy, milky substance that comes out of the mushroom as you damage it. And it is definitely, in the case of both of those species, it's sticky, it's pungent and funky. Like it has a fairly unpleasant, like underwear adjacent aroma. It's not great. And so like when you start to handle these mushrooms, they may not seem terribly appealing for the dinner table, but when they're cooked, they actually have a very, very nice fish meaty sort of thing going on. And but not fishy fish, like fish as in a very nice sort of meaty piece of fish, like a tuna or something similar. So as I'm like waving this around, you can see little droplets of this latex just going everywhere. And as the mushroom sort of like that damage has a little bit of time to settle, it turns a dark brownish color. It's almost black. And so the gills are sort of this like pale, they can start out almost creamy color, but by the time they are fully mature, it's almost like a creamy orange tan is sort of the, the color of the undamaged gills. And I'm gonna drink some water here. One of the things I will note is, again, the conditions have been quite dry this last week. And so even though mushroom hunting is a fairly like low key, it is not cardio. It is definitely a season where I have had to spend a lot more time reminding myself to hydrate. All right, so remember to drink water. I am full of really valuable information today. So let's return to our Lactifluus corrugis. So you've got all of this, just tons of milk going on in the corrugated cap. So I've really covered the identification features for the most part. I think the thing that is really enjoyable to me also about this mushroom and Lactifluus volemus is that they have these very beautiful stems as well, and they're smooth and sort of colorful. And so they almost have a nice, beautiful candle type, like waxy candle type of appearance and feel. So like the aesthetic and the vibe of these mushrooms really suits me. Again, the aroma, not great when they're fresh. Certainly the darkening color of this milk can also be a bit of a turnoff. But I do very highly recommend that, you wash them thoroughly and cook them thoroughly, and then they are a very, very nice sort of seafood, like, meaty fish type of mushroom food. Okay, so I also want to talk very briefly about, the family that this mushroom occupies. So I mentioned that there are 3 genera of mushrooms that are milky mushrooms. So you have Lactifluus, which are these dudes. You have Lactarius, which is a much larger genus and is much more familiar to many people. So, originally Lactifluus volemus and Lactifluus corrugis were Lactarius volemus and corrugis respectively. But Lactifluus has been split off, and it's a lot of mushroom species that are like tropical and subtropical. But there are, I guess, ostensibly many of them. So, north of 100 species. And, the ones that I see most typically besides the volemus is Lactifluus piperatus, which similarly is a milky mushroom that produces a lot of milk from its gills. And so, some of the other milky mushrooms in the Lactarius genus, they produce milk, but it's like sometimes it's scant or it's watery. But in the case of all of the, the peppery milky caps and the 2, the fishy milky cap and the corrugated fish milky cap, they all are Lactifluus and they all produce a lot of milk for me. So that's one of the ways that I remember them. But it is kind of peculiar to me that this mushroom is listed as growing in association with oak. And, we collected them in an oak, like a mixed forest. There was a lot of rhododendron, but certainly I think it is, it makes sense that it was growing with oak. And I just am surrounded by those kinds of trees, and I just don't find this particular mushroom. And I think that's one of the reasons that the observational science of mycology can be so rich and rewarding. And, as far as the methods that people use and how we're refining understanding the range of different species is the area I think I'm the most interested in. So if you've watched this channel before, there was a couple of months ago, I found a bolete type mushroom, a Tylopilus mushroom technically. So it's like this reddish purple bubble of joy. And it was Tylopilus williamsi, or at least I got, very good verified identification from experts that that's what that was. And that mushroom does not, occur in Raleigh, North Carolina. It's listed much more as a Texas and far more southerly species. And so when it comes to these sort of large regional groupings and where things live and where they don't live and why, that's an area where I find there to be a lot of mystery, but also a lot of our data gathering techniques, I think, are getting us to an area where, as sort of hobbyists or people who are approaching macro fungi at a macro level, going out and picking mushrooms as opposed to running DNA sequencing or doing microscope work on everything we find, a lot of the methods we have for sort of understanding where stuff lives is getting much better. And to that point, I wanted to share sort of a couple of the methods that people are using and talking about. And again, I am not an expert, and I highly recommend that you look into the Fungal Diversity Project on iNaturalist. And so that is one of the projects where the cataloging of different organisms that are found is done. We also have have, myco blitzes that are organized, and most of these observations are gathered through the application iNaturalist, and then folks contribute also fruiting bodies that can be DNA sequenced, so we can do these sort of like, distributed online data gathering sort of exercises that help us understand, for instance, I don't know why I don't find this mushroom in Raleigh, North Carolina if it grows with oak, but maybe someday we'll have enough information to know if my neighbors experience that. Like, as of now, all I know is I've talked to a couple of other Piedmont mushroom hunters, and they're like, you know what, you're right, I don't find Lactifluus corrugis down in the flatlands. I find it up in the mountains, but sure enough, we don't have enough information to know for sure. Okay, so the Fungal Diversity Project is really what you kind of want to investigate, and there's a lot of really good resources about how to document your finds on iNaturalist, and get involved also with what I was just talking about, myco blitzes, which are essentially, a period of time where people can gather specimens and then prepare them and send them for DNA sequencing. So that's very fancy and a very good way to sort of get more information. So all of the sort of cataloging of, mushrooms in these more regional units, like that's the thing I'm very interested in. I really enjoy going to a foray and doing sort of what is called a survey of alpha diversity, meaning the diversity in one very specific site and all the different species in the places where you have a group of people collecting, identifying, and cataloging everything. And as you descend through the Greek alphabet, you get to sort of larger and larger units of, where you're assessing diversity from. And once you get to the place of epsilon diversity, and I saw this marvelous talk by Stephen Russell, an academic from Indiana who works on mycoblitzes and fungal diversity, and does a lot of this sort of cataloging I'm talking about, assesses epsilon diversity, which really is a region that is large enough that you can talk about as a distributed human community of mushroom hunters. So in Stephen Russell's case, that is, the macrofungi or the mushrooms of Indiana. And so what they do in terms of trying to understand this epsilon diversity, so it's like, I'm not talking about what I see at this particular location. I want to understand how many milky mushrooms do we have in the state of Indiana. And that's a really good, given how rapidly we as people can move around and how much we can explore, those kind of larger, units of understanding diversity is very helpful. All right, I'm going to drink a little bit more water and talk about just a couple of the dimensions upon which these sort of cataloging exercises, rely. And most of this is in the context of like citizen science with a very, very small, but, dedicated academic community that's distributed across a lot of different institutions. But you have a lot of, again, citizen science and people contributing observations and their volunteer time looking at and processing people's specimens. Okay, so if you're looking at epsilon diversity, if you were, say, looking at the diversity of the Piedmont region, so that's where I live, I call it down in the flatlands. So you have the Appalachian Mountains, and then you have a big corridor of land that is, has a lot of broadleaf trees, a lot of similar habitats, but a lot of different mushrooms. So if you're trying to figure out, what lives there, and you are deploying people, obviously you're going to be asking them to, collect high quality specimens of mushrooms. And so, the first step in doing this, if you want to contribute, is to, take photographs and publish them to iNaturalist, and you can get help with identification. And that's a really good way to kind of get your feet wet with, providing photographic evidence to this distributed community that, yes, I found this thing in this particular area. And, iNaturalist does provide, geotagging and information that gives sort of geographic, information and data, metadata, to folks who want to do research. So that's thing one. Besides those observations, we have these structured collecting exercises, for instance, the foray I'm attending. So each of the different specimens that people have collected that we are taking to, get identified, we are, and when I say we, I mean a very dedicated group of very smarty pants volunteers, are filling out vouchers that basically document some of the information about the mushroom, where it was found, who the person is, who collected it, and some of the field notes, and then, cataloging everything. And then additionally, a lot of those mushrooms, or at least, some of those fruiting bodies will be dehydrated, and there will be DNA sequencing done on them using a method that's called nanopore sequencing. And so that is one of those, I think, things I wanted to mention as far as these cataloging exercises. We've had a lot of mushroom forays for a very long time, and you'd have people, collect, dehydrate, put things into, various herbaria or fungaria that are, at academic institutions, and oftentimes there would be microscope work done, but we also have this additional dimension of being able to get some, genetic analysis done of things. So you have a variety of different sort of data inputs besides just the fruiting body. You have some genetic information, you have geographical information. A lot of times you have folks who are doing, microscopic photography, so you get images of things like the structures on these gills. And, the gills are this whole, really elaborate structure for creating a place for the spores of the mushroom to develop and then be dispersed into the airflows. And so there's a lot of really interesting and distinguishing stuff that happens there. So you have all of that information that comes together. And that gives people who are looking at these wider units, these epsilon or larger units, or smaller, an opportunity to say, okay, this is the number of species that we think that resides within a certain area or a certain zone. And another method that I actually am just learned about in the lecture that I was just talking about with Stephen Russell is doing soil sampling as well. And so sampling soil for mycelia and other fungi, and so using that as another stream of data to contribute to these observations that are done of actually collecting fruiting bodies and running genetic analyses. And the long and short of it is, of course, the more we go into these things, I find it to be deeply fascinating. And the answer is almost always there are more mushrooms, there are more species. And so, one of the things I really enjoyed about that lecture is that looking at the macrofungi, at the mushrooms of Indiana, they're starting to see in the amount of data they're gathering a hint of a plateau in certain areas of mushrooms that are highly conspicuous. So for instance, milky mushrooms, there's a lot of different people who've been contributing, specimens, and they're looking at, I think it's somewhere in the 70-ish region of mushrooms in the Lactarius genus. And so as time has gone on, it's okay, we know that there are 50, and now we're at 65, and they're in the 70s, but they're starting to see, again, with a lot of hands and eyes and soil and DNA, a chance that we have a sense of what mushrooms live where, and that's very helpful, at least to somebody who's a hobbyist like me. Anyway, I've gone on at great length, but suffice it to say, Lactifluus corrugis, the corrugated milky cap, is one of the reasons that I like to travel and go up into the Appalachian Mountains. And so that plus mushroom festivals. Oh, and plus also actually Sewillis spraguei, the painted Sewillis, which is just a gorgeous mushroom that also it grows with white pine, and we don't have white pine down the hill in my Epsilon region. And so coming up here is an opportunity to see a mushroom that is more Dr. Seuss-esque and has this wonderful sort of radially aligned, honeycombed, spongy undersurface and floofy, nice, sort of nice mulberry red cap. I just don't get to see that at home. So, everyone has a different reason for going on adventures and traveling, and for me, milky mushrooms and mushroom nerds is more than enough. I hope you're doing great. Find a billion mushrooms. We'll talk again soon, I hope.